Hello and welcome to today's webinar. On behalf of Drug Target Review and SPT LabTech, I'd like to thank you all for attending. I'm your moderator, Ria Kakad, Editorial Assistant at Drug Target Review. Today's speakers will be Dr. Hugh Rees, Field Application Scientist at SPT LabTech, and also by Susmith Mukund, Principal Scientist at Frontier Medicine. Following their presentation, we will move on to a live question and answer session, where you can ask any question to today's speakers. Please remember, you can submit questions at any point during the webinar using the question panel situated in the menu on the right. So without hesitation, may I pass over to today's speakers.
Urea.
Thank you.
So for today's presentation, I'm going to be discussing SPT's liquid handling instrumentation and how they can assist your automation for structural biology, particularly crystallography. First, I'll go through and demonstrate discuss a workflow for crystallography with these instruments. Then I will introduce each instrument in turn. And then eventually I will pass on to my collaborator Susmith, who will discuss his use of these instruments. So just some quick background about SPT LabTech. SPT LabTech have been in the industry for a few decades in liquid handling, structural biology, and sample management. We have a reputation for designing and manufacturing robust, reliable, and easy to use instrumentation for sample management, structural biology, including now cryo-EM, and liquid handling across many volume ranges. Our goal has always been to enable life scientists through collaboration, deep application knowledge, and leading engineering to accelerate research and make a difference together. So, 1 second. So just a quick introduction to me again. Thank you, Ria, for introducing me earlier. I am a field application scientist at SPT LabTech. I'm based in Boston and I service the Northeastern Territories in the US. I've been with SPT LabTech since April 2022, and I am experienced with automated workflows in crystallography. In fact, my background was using a Mosquito for setting up crystal trials and crystal trays. I've also, however, learned with SPT LabTech how to use SPT's liquid handling instrumentation for genomics applications, including library prep and miniaturization of library prep, and also drug discovery and high throughput screening. Whoops, sorry, I skipped over that slide. So just to recap today's discussion, I will be discussing 3 instruments that SPT LabTech makes and services, and explaining how they can assist you with automating your crystallographic workflows. The first instrument, I want to introduce the Mosquito Crystal 3, which is the picture on the top right. This is one of our most steadfast and reliable instruments that's been around for a while and is extremely well known for setting up and managing very small amounts of liquid in the sub-microliter range. You can set up crystal trials in sitting drop, hanging drop, and other variants of crystal trays very easily in a miniaturized volume. The next instrument I want to introduce is the Apricot S3. SPT LabTech acquired Apricot Designs in 2021 and integrated their portfolio into ours. The Apricot S3 is a reliable plate stamping instrument that can be used for serial dilution as well. It is an air pipetter, but has a very good performance with many liquid classes anyway. The third instrument I'd like to introduce is the Dragonfly Crystal, which is the picture on the top left. This instrument is a bulk dispenser specialized in creating plates with up to 10 different components in various different gradients and concentrations, and is ideal for setting up customized plates of condition for crystallography. So to demonstrate how these 3 instruments integrate together into a workflow for crystallography labs, I'm going to show you a video where I physically use them and show how they work together. So here I am using the Apricot S3 to transfer crystal conditions out of a Hampton deep well block into a sitting drop tray ready for drop setting at a later point. So you simply load the tips into the Apricot and you could easily program a simple mix and then transfer out of the Hampton deep well block into a sitting drop tray. By using automation, you make this step consistent and reliable. Now we move on to the Mosquito, where we're going to do our drop setting, where the tips aspirate from the conditioned wells and drop onto the protein, which is deposited by the tips in the earlier step. And then we use the Dragonfly to create the optimization screen. So after you've done the crystal trial, grown the crystals, looked at them, if you need to optimize beyond that point, the Dragonfly is perfect for creating those much narrower range of condition space that you need. You can then continue to use the Apricot to transfer out of those sorts of plates back into sitting drop and repeating the crystallography cycle once more. So now that we've watched that video, I'm going to break it down in a more kind of picture format of what was going on. So in a typical crystallography lab setting, you have a protein or macromolecule of interest, shown on the left here. And to test in what conditions that will crystallize, we almost always start with a commercial condition screen, such as the one here shown here from Hampton Research. These deep well blocks contain 96 different conditions with various components, salts, buffers, and so on. And we need to transfer those onto plates where we have a drop of protein and a drop of the condition screen to combine and create the conditions under which the crystal will grow. This can all be done by hand, but in this situation, normally you would have to use about a microliter of protein and a microliter of condition screen to create a drop. You could go lower, but by hand it's very difficult to accurately pipette those volumes, especially if they're very viscous or have other unfavorable properties for air displacement hand pipetting. Once you've set up these drops from the normal commercial condition screens, you can see the crystals. Either you grow good crystals, you get the data you need for a structure, and that's that, or you grow poor crystals or ones that don't quite give you the right information. And here you need to optimize. So for optimized trays, you need to create your own conditions, usually using components sold by Hampton, but you still need to nevertheless create that very narrow pH range or salt range that you need to probe what will create the best crystal. This then goes back into the condition screening cycle, and therefore the crystallography workflow cycle is complete. So as I mentioned, all of this can be done by hand, but protein needs to be very concentrated for these crystal trials to work, and making enough protein and purifying enough protein to get to this point means it can be exhausted very quickly if you're only testing one condition per microliter. This is where the first instrument I'm going to talk about, the Mosquito Crystal 3, comes in. The Mosquito Crystal 3 is essential for setting up miniaturized drops of protein and crystal condition. The Mosquito can set up with a humidity chamber drops of 100 nanoliters plus 100 nanoliters of protein and crystal, and it can set more than one drop per well condition, as shown here, such as 2. And therefore it can screen a vastly greater number of conditions than you would ever be able to screen if you were to do this all by hand. The Mosquito is essential for not only getting a massive throughput of crystal trials, but also miniaturizing and saving that precious protein or other macromolecules that you make and purify to get to the stage of your research. However, the Mosquito is not able to work from a deep well block like the Hampton screen here. So to make sure you're transferring these Hampton screens consistently out of these deep well blocks and into these sitting drop trays, the instrument we suggest for that from the SPT range is the Apricot S3. The Apricot S3 is an air displacement pipetter, which is able to aspirate from these deep well blocks and also importantly, mix them first before transferring into a sitting drop plate like this one here. By using an Apricot, you make the process consistent, reliable, and essentially reduce the risk of contamination or mishandling, which can happen by trying to pipette these each in a multi-channel or one well at a time by hand. The third instrument I want to introduce from SPT LabTech again is the Dragonfly Crystal, which is focused on this customized condition step. When you have a crystal condition, but it has pH you want to vary the pH or the salts or the buffers, this is where the Dragonfly Crystal shines. It is able to assemble these plates easily, reliably, and you are able to design them in ways that you simply could not by hand due to the great range of its dispense capabilities. So these three instruments together massively automate, increase the throughput, and most importantly, from a crystallographer's perspective, make it much more reliable and consistent in your approach to crystallography. First instrument I'm going to talk about in detail is the Mosquito Crystal 3, which is the drop setting instrument for crystallization trials. So just to go over some specifications and overview, the Mosquito Crystal 3 has 8 channels, 8 tips below the head, which can aspirate, mix, or dispense from heads. The plates on this deck, there are 3 plate positions on a Mosquito Crystal 3 deck, use the standard SBS format, and they have 2 protein strips between the first and third plates, which contain very low dead volume plastic strips, which can hold protein or other very precious reagent that the mosquito can aspirate from. The mosquito is able to work with various heights of plates, it's able to work with various shapes of wells, and it's very easy to program. The mosquito has a completely user-friendly UI, which requires no programming experience. We do have the option of using CSVs or work lists, as we call them, to program more sophisticated or modular program protocols, but this is not necessary to get most of the use out of the mosquito. The mosquito, although it only works 1 column at a time, is a very fast instrument and is able to copy a 96 well plate in roughly 2 minutes. It is a highly accurate instrument thanks to its positive displacement, which I will talk more about in a moment, and it's got very reliable and repeatable transfers of virtually any liquid. Just to explain why the mosquito is so accurate and so reliable at such low volumes, I'm going to explain the positive displacement pipetting technology at the core of the mosquito. The mosquito tips are composed of a sort of tape which was mounted on that spool at the back of the top of the instrument. Each tip is composed of a steel piston, shown here, inside a high-density polyethylene barrel on a polyester backing tape, which is run through these holes like a ticker tape along the machine. It uses positive displacement, so there is no air gap between the liquid and the steel piston. It's accurate for high viscosity, dense, surfactant, volatile, hot or cold liquids, because this steel piston displacing the liquid means it's very unlikely that it's going to aspirate the wrong volume. There's no air, there's nowhere else for the liquid to go, so it has to go inside the tip. Because of the positive displacement, it also has a very low and very small dead volume in the range of 10 or less nanoliters. It cannot be blocked because the pistons you move with enough force that any precipitant or otherwise viscous reagent will leave the tip, it will not be stuck. However, despite having a steel tip and powerful movement of the piston, it exerts minimal shearing and frothing forces because there's no vortex point at the end of the tip. So it's very gentle on your macromolecules. They are also disposable. Mosquito tips travel through the machine on a one-way track, so once they are used, they leave the machine. There is no chance of cross-contamination. And you don't need to replenish them very often, as there are up to 36,000 tips on one spool. In this video, you're going to see the mosquito tips aspirate this blue liquid and dispense it onto a plate seal. You're then going to see it aspirate more blue liquid and mix it with this yellow liquid here. I'm just going to pause it there because I think it's going to go to the next slide here. So sorry, just to show that again. So the mosquito is able to aspirate from this plate, it's able to dispense, and it's able to mix all with these steel piston tips. It's showing 16 tips right now, which is another option for the mosquito, but generally 8 is what we would use with the crystallography. Although the mosquito hardware has been around for a little while and it's very reliable, the software has been evolving as time's gone on. These days, Mosquito Crystal 3 software is the most useful it's ever been for helping crystallographers set up their crystal trays. When you open Mosquito Crystal 3 software, the modern version of it, you are met with several options for running all sorts of crystallography experiments. We have folders for sitting drop, hanging drop, and microbatch, which each contain example protocols or which are fully functional for each of these processes. We also contain options for showing you how to use the mosquito for bi-cell substrate 96, soaking, crystallization, microseeding, other optimization processes, as well as additive screening. We do have the old school protocol tab where you can write your protocols one step at a time, as has always been the case in the mosquito, but these experiment tabs help you write experiments more easily by having a much easier to use starting point or a way to help you visualize how the mosquito can be best used for what you need to get done. Next slide, I'm going to focus, for example, on what we suggest for the sitting drop trays in the mosquito. So for those of you who don't do sitting drop, I myself was a hanging drop person for many years. The sitting drop wells generally work in this basis where you have the well condition at the bottom of these large wells, and then you have a sub well within the same well on the 96 plate, if you like, where you will drop the protein and the crystal condition. Using the vapor diffusion effect at the heart of a lot of crystallography, at the heart of hanging drop and sitting drop, we are able to grow the crystal in this subwell here and keep the environment stabilized by having access to the well condition here. So in the Mosquito, if you go under the sitting drop templates, we have an example where one drop, we have a example template experiment where you set up you set one total drop with 2 drops from 100 nanoliters of protein and 100 nanoliters of condition inside each of these subwells. We also have templates for setting multiple drops, like on this tray here, which has up to 3 subwell drops per well, per large well. And you can change and modify what volumes you are using for these drops very easily in the software. You can do 100 plus 100, you can do 150 plus 150. Sorry, one second. Sorry, what the next slide was. The main thing to note, though, is that the Mosquito is not able to set up these well conditions inside these large wells by itself. Although it's perfect for setting up these sub-well conditions and actually starting the crystal trials, to get these conditions into these wells, you either have to use a handheld pipette or a plate stamping technology. And the technology we have available from SPT LabTech to do that is the Apricot S3. So as I mentioned earlier, SPT LabTech acquired Apricot Designs in 2021, and part of their portfolio was this extremely reliable air displacement pipetting head, which we have now integrated into our other systems. But the one here is the Apricot S3 with these 3 plate deck positions and this one pipetting head shown here. This video will give you some insight into how the Apricot S3 works. It has text, so I'm going to avoid talking too much so that you can read what it's saying. Second. Unlike the Mosquito, the Apricot is able to work with deep well blocks, which means it can be used with Hampton blocks. Pipetting core allows you to switch between 96 and 384 if you desire. It's an extremely easy to use instrument, which also doesn't require any coding experience. The tips are loaded very easily on a tip clamp. These are proprietary tips, so we don't have supply chain issues. And with a deep well block, you will be able to create 2 sitting drop or hanging drop trays at a time very easily using the Apricot S3. Just to pause here, the volume ranges, oops, that stopped at the end of the video. Get to the volume ranges, sorry. So the volume ranges of the Apricot depend on the core and tips that are loaded and the tips being used, but essentially it can go from 0.5 to 1000 microliters, meaning it accesses a range of volume which the Mosquito doesn't normally access, making it an excellent complement to that machine. It is also able to work between 96 and 384. With full head pipetting for plate stamping, it is able to use single columns as well, so it is able to serially dilute. You have filtered and sterile options to keep your crystallography workflow clean. And it can fill, copy, reformat, and work between all sorts of plate formats. The core at the heart of the instrument is user exchangeable, so you can switch between 96 and 384 in seconds. The tips are easy to load, and there is an assured tip supply chain. Another key thing about the tips is the way they seal against the gasket inside the head means there is even pressure and force exerted across the all 96 or 384 tips. It's not like deformation seal tips where the ones at the edges or the ones in the center might be better or worse sealed than the others around it. The Apricot provides a very even and reliable sealing across all tips. Because you can partially load the head, because of this, you can also partially load the head in many different arrangements and still see the same effective pipetting functions. and you can reformat between 96, 384 fill plates, empty plates, and replicate plates very easily. We also do have the option of doing serial dilution on the Mosquito with a single, Apricot with a single column. And the software is very intuitive with a large touchscreen interface, which is very friendly with for new users. It also has a very small footprint, making it easy to fit into your lab space, no matter how compact it is. The next instrument I'm going to talk about is going to be the Dragonfly Crystal. Dragonfly Crystal is perfect for creating optimizations of condition screens that you find through your crystallography. So although it isn't really used for the first cycle of your crystal trial workflow, it can be extremely important for the steps after, particularly if you're trying to lower the resolution of those crystals you do have, or you're trying to find a way to get your macromolecule to crystallize in a way which produces the data you need. So the Dragonfly Crystal also uses positive displacement like the Mosquito, but it uses a completely different shaped tip. On the right, we have a syringe. This high-density polyethylene barrel, this plastic barrel, polypropylene barrel with a high-density polyethylene plunger forms a deformation seal, which is the positive displacement technology interface, which we use to aspirate from these reservoirs shown here. You can have up to 5 or 10 different heads on a Dragonfly. Each head is able to aspirate and dispense independently of each other. When I say head, I mean this steel piston coming down from inside this white box into these syringes to do the aspiration and dispense. The syringes, just when they do dispense, are non-contact, so they are above the plate and they do not go inside or in the well or come into contact with anything inside the well. So they are very easy to keep clean and very reliable at not contaminating your plates. Are very fast. A well-equipped Dragonfly with multiple syringes for a component can fill a 384 well plate in around 30 seconds. The volume range of a Dragonfly Crystal for dispensing is from 500 nanoliters up to 4 mils at a time, with 500 nanoliters being the minimum reliable volume of dispensing and 4 mils being the full volume of one of these syringes. These syringes aspirate from reservoirs containing up to 10 mils. The Dragonfly is gentle enough for cell dispensing and other substances which do not respond well to forceful pipetting, and it is very accurate and reliable. The plates that you want to fill fit into this slot here, which moves under these syringes to fill across the plate. The reason for the Dragonfly's inception was that it was a lot of optimization in crystal trials are, even if they're done originally on the Mosquito, then done by these larger, less efficient plate formats such as 24. This is generally done to grow larger crystals, but it does slow down the process an awful lot. So before the Dragonfly Crystal came about, this was mainly done by hand, and it was a very long and slow process for any crystallography lab. With the Dragonfly Crystal, you can have 5 or 10 heads, which can hold up to 5 or 10 different components, with these disposable syringes that minimize the amount of time you have to spend washing or cleaning. And these syringes can dispense different liquids with different volumes very easily and reliably. In this video, you're going to see this Dragonfly dispense ethanol and glycerol at different volumes adjacent to each other. Note that because of the positive displacement, the tips don't clog. They are unlikely to ever get blocked, and they're very reliable for dispensing a huge range of different liquids. And MPD and glycerol and ethanol are all possible components that you'll be using in crystal trials. The resolution and precision of the Dragonfly is exceptional. With a hand pipette, accessing accurately below a microliter is very difficult. And although you might be able to do this if you're focused once or twice, relying on this happening time and time again, as you set up plate after plate, isn't really realistic. The Dragonfly is able to set these plates up reliably and consistently time and time again. And also, because of its 500 nanoliter minimum dispense, it is able to reliably access a range of different concentrations and buffers that you could not by hand. With a Dragonfly, you need fewer stocks of these sorts of components because you can have a more concentrated stock, and the Dragonfly will be able to do the small piece dispense, which saves you that material in the long run. Setting up these plates, it's extremely reliable across its dispense range from 4 mil to 0.5. It's very accurate, and it has very little problem, as I said, with different types of liquids. It's also able to dispense different volumes in each well. It's able to do gradients. It's able to dispense one component, one column. Anything, any arrangement you could think of, the Dragonfly is going to be able to do that. To assist users with creating these plates that might be very complicated and have different gradients and different columns, we have a dedicated design software which facilitates the creation of the files that are used to run the Dragonfly, in addition to the simple plate fills which are possible to do on the baseline Dragonfly software. Here in the designer software, we have, for example, these two buffers and two components, and we are specifying the different concentrations they are across the plate. We're specifying the type of gradients they are using, and we can add up to 10 different components for a 10-head Dragonfly here very easily if we wish. The Dragonfly can do these, generates then a CSV file. The software generates a CSV file which can be loaded into the Dragonfly for easy use. And these are very modifiable, and you can create whatever sorts of gradients or crystal conditions you are interested in. It's a very easy instrument to use. Within a minute, you can go from putting your syringes in, you can put the file in and then hit run, and it will engage the syringes, aspirate, and then fill your plate. It's very, very hard to go wrong with a Dragonfly in terms of using it correctly. The tips go in, you add the plate, you add the stocks, and that is all you have to worry about. So the Dragonfly is really helpful for setting up these sorts of plates, and that really helps for creating customized crystal conditions because it can deal with any sort of liquid class. It is very good at being repeatable and reproducible, and also the force at which the Dragonfly exerts its dispensers into a well can mix the conditions very easily. There is virtually minimal risk of contamination because the Dragonfly is a non-contact dispenser and dispensing into the wells from above. And it has very good resolution across its entire dispensing range. It is also a relatively simple instrument to use. When I go into a lab setting up a demo for the Dragonfly, it can go from being from off the bench onto the bench and working within minutes. It's an extremely robust and reliable instrument, making it very popular in the lab beyond its base applications. So now I'm just going to talk in detail about how these three instruments will work together and create the crystallography workflows you need. So for initial screening, the Apricot S3 is essentially your best instrument for transferring out of your deep well blocks, whether they be Hampton deep commercial blocks or other commercial conditions that you've set up into into a deep well tray. The Apricot S3 is able to transfer into hanging drop plates. It's able to transfer into sitting drop plates, and able to transfer into all sorts of different plates. It's very easy to train a new plate on the Apricot, whether you're using brands like Intelli or you're using the SBT brand or anything else. The Apricot is very adept at working with all of these different kinds of plates. In terms of keeping the tips clean for doing those transfers outside of those deep well blocks and things like that, you have options such as pre and post air gaps. We have tip touchers where it will touch the tips to the surface of the liquid and then to the side to make sure that everything gets left behind, keeping the tips clean and ensuring there's no contamination. And it can also set up 2 of these plates at a time, meaning that it's slightly more efficient. And it's still a fairly hands-on instrument, but it's very easy to use. And because of the plate stamping and serial dilution having applications well beyond crystallography, it's an extremely versatile and affordable instrument. Mosquito's drop setting, as I mentioned earlier, it can do all sorts of different drop setting scenarios, such as sitting drop, hanging drop. It can also do microbatch under oil. It can work in 96, 384, and 1536 formats. And essentially, the only drawback to the Mosquito in this respect is it cannot really work with deep well blocks. So with the Apricot as a companion to the Mosquito, you are able to transfer out of Hampton deep well blocks, out of larger condition plates that you might use as your stock, and transfer into the plates that you will use on the Mosquito immediately before the experiment. So you aren't having to prepare all of these different plates. You can just go into the workflow and get your crystallography drop setting started all in one lab session. Once the Mosquito has set up the plates, you can then seal them very easily and study them for crystal hits. Depending on how you do that, you might use an imager, you might do it by hand, but ultimately the crystal, even if you aren't necessarily using the most out of the throughput, speed of the Mosquito, you are still using its ability to reliably set up these miniaturized drops for your crystallography, which is a very important part of any crystallography lab's workflow and frankly time and finances, because you are able to save your protein and you're able to save those very precious conditions that you're using to set up your crystal trials. Once you have crystal hits, whether they be diffracting or not, and you want to optimize, the Dragonfly Crystal is really the next step in the process. So as I've mentioned all before, you can use this to set up the plates that will optimize these conditions with variable pHs, salts, and all buffers and all the other components you might need in the crystal trial. And they can set up these plates very quickly. You could make you can make 4 or 5 different optimization plates in a matter of like 15 to 20 minutes if you wanted to, and you can test all of those on the Mosquito very easily. Once you've created these plates, you could set them up in slightly larger plates, like a 1 mil condition plate, and then stamp them out from an apricot, so you are using the same source plates each time. Essentially, because you're making fewer plates, you have less variability between them, and using the apricot to mix and aspirate out each time ensures you're not not changing the condition over time because there was some precipitate in there or other things. So the Dragonfly, apricot, and apricot do work together for the optimization stages of your crystal workflow, and then you can use the Mosquito once again for drop setting and getting your crystal hits. So without further ado, I am going to head to my collaborator, Susmith.
Thank you, Hugh, for the nice presentation on what these instruments can do. So hello, everybody. Once again, my name is Susmith, and I'm presently a principal scientist in the structure biology group at Frontier Medicines. And I've been fortunate enough to have a wide range of experience in structure biology at some really good industry companies like Gilead, Genentech, and Amgen more specifically. And through the course of my studies and discoveries, I've had fortunately a rather successful track record tackling these tough problems. And I just want to kind of present a use case scenario here, continuing upon what Dr. Rees just presented. Next slide, please. So I think you skipped a slide there, Hugh. So as most of us may be aware, I just wanted to start by presenting a general crystallography workflow. And as we know, we need really highly pure and homogeneous pool of high quality protein for crystallization efforts. And we need to be able to concentrate it sometimes to pretty high amounts. And the traditional way to set up was to use older methods by hand. But in the last couple decades or more, we have been fortunate enough to use several robotic instrumentations to make this process more feasible and actually delve into more high throughput screening modalities. Of course, following successful crystallization experiments, we would basically screen for crystals using either any microscopes or imagers, and then fish the crystals and try to shoot them either at an in-house source or at a synchrotron, collect data. Of course, integral to the part of finding hits is also optimizing the hits, and we would today in the present scenario use some of these robotic instruments as opposed to just using manual methodologies. Following achieving successful crystallization and optimization, we would need to then obtain high resolution data sets and then solve these crystal structures to hopefully keep the cycle of structure-based drug discovery going on in the industry. Next slide, please. So why then would we want to try to do this in a very integrated manner using robotics? So as I already alluded to, you want to try to come up with a very simple and an elegant process to purify protein to homogeneity, concentrate it, and prepare your protein sample for crystallization. So once we had this, next slide, please. There's a few animation slides. Yeah. We want to arrive upon then deciding on the screens that we want to use. And as I alluded to, and as Dr. Rees alluded to, some of the attributes we want is basically to make sure that our transfer from these 96 plate screens to the crystal setup 96 plate wells need to be very consistent and reproducible across all kinds of solvent and liquids, regardless of the viscosity. We also want to minimize the time that we are trying to do this, because not in all cases you may not have your protein that is really stable and being able to sit forever on ice. So considering all these attributes, one of the solutions that I arrived upon was a liquid handling system like the Apricot S3. Next slide, please. To actually facilitate this transfer and to remove any problems we would have otherwise, as in fatigue, to remove errors and waste, repetitive motion injuries, and inconsistency caused due to manual pipetting errors or calibration issues or viscosity issues, and to do this in a very shorter time frame. Please, next slide, please. To address all these issues, I found the Apricot S3 as a use case, in this use case scenario that I'm presenting, to be very useful, and it also addressed all the attributes that I actually was looking at to try and solve this issue. Next slide, please. So now, once we have your plates actually stamped using this particular robotic instrument, which I consider to be very elegant and simple way to solve any issues, you can actually transfer 96 well plate into your crystallography setup. The next step is to go into setting up your protein in the crystallization experiments on your crystal plates. And for this, I actually relied on the Mosquito X3. As Dr. Rees has alluded to, the positive displacement tips are highly accurate and deliver reproducible volumes in a very highly repetitive manner. You can set up very low volumes and fast, and I've actually timed this over the course of my experience. You can set up one protein in a 96 well tray in under 4 minutes, and you can also set up multiple drops of the same protein or multiple drops of 2 different proteins mixed using these mosquitoes in as little as 5 to 7 minutes, depending on the plates you're using. Of course, I don't recommend trying to set up 3 drops or 3 different protein in one shot, because you still want to avoid leaving your protein, especially in these small volumes, out exposed to air for a long time. But the other solution to that SPT LabTech and this Mosquito X3 and all other similar mosquitoes provide is a built-in humidity chamber, which you can control to be set at a desired humidity level, so you can reduce static evaporation loss from these 100 to 250 nanoliter drops that you're setting up in these experiments. So this would be a very simple, very elegant, and an ideal workflow, which you can consistently set up in a very fast manner and also in a high throughput fashion to actually screen a large array of conditions for your particular protein of interest. Next slide, please. So some of the advantages to this very simple crystallography workflow, as you can all imagine, we put in a lot of effort today to get these targets ready for crystallization. And among all the targets that are present out there, the ones that we are working on today have become even more challenging and tough to make. And so we don't often get easy to handle proteins anymore. We don't have the luxury of making large amounts of these multi-protein complexes anymore. So miniaturizing the system and trying to screen a wide array of conditions, especially if you're able to only get very little protein, so setting up 100 nanoliter drops and searching a larger crystallization space, with the amount of screens that are available today in the industry, makes it actually possible to get hits for these tough systems. Next point, please. Again, as a corollary to this, time is also of essence in our highly competitive industry landscape. Whether you are in a continuous crystallography lab or a lab operating around scheduled beam times, we have these elaborate workflow processes for these tough targets, and these things can be made consistent, very simple, and reproducible to achieve success. Next point, please. Of course, being able to include automation is a very critical part of this workflow in today's crystallography space. This I found, and as most of you might be aware, enables consistency and rigidity in several of the critical screening experiments. It certainly removes user error, as you are all aware, that can be caused by several reasons. It improves the timescales for experimental setup, and it keeps it consistent across the board. And more importantly, it also ensures any sample waste, especially I'm sure that most of you have handled proteins at, as low as 50 microliters that you want to set up to try to get hits. The other part of actually, once we get the crystal hits, what are we going to do? And this brings me to basically another robotic instrument that was addressed here by Dr. Rees to potentially add on very significantly to the productivity workflow. So of course, you are going to set up a whole bunch of these sparse matrix screens, and once you arrive at a hit, one of the standard procedures is to make grids to optimize the hit. And the standard process is, of course, to try make 24 well plates, or I've even made 48 well grid plates. But next point, please. If you're able to integrate a robotic liquid handler like the Dragonfly Crystal, this makes this process very straightforward and incredibly easy. And I found it to be very useful and very valuable in order to make several such grids for screening and elaborating on my grid following a potential hit. I've also found the Dragonfly Crystal to be useful to make master plates in order to do repetitive screens. So you can design your own grid as well and use it ad infinitum using basically the Apricot, which is a plate stamper essentially. Next slide, please. So with this, I'll hand over to the organizers to answer any questions.
Thank you, Susmith.
Thank you both. Thank you, Hugh. Thank you, Susmith, for your great presentation. We now have the pleasure to introduce our question and answer session, where today's speakers will answer all your questions. Don't forget, you can still submit your questions using the question panel in the menu on the right. So let's take a look at our first question. Firstly, how much do these instruments assist with miniaturizing crystallography?
Yeah. Okay. Yeah. So these instruments primarily help with miniaturizing crystallography at the Mosquito step. Mosquito is able to set up drops, as Susmith has mentioned, down to 100 very reliably. It can also go even lower, but even at 100 or even 200 nanoliters, it's still using either 10 or 5 times less than you would be doing by hand with a 1 microliter instrument. So generally, that's the range we recommend going down to with the Mosquito. It does, the minimum aspiration dispenser Mosquito can do is 25 nanoliters, but as Susmith has also mentioned, we wouldn't recommend going that low. The Dragonfly doesn't really help with miniaturization of crystallography, but it certainly does help expand the range of conditions you can test. And the Apricot simply aspirates and dispenses very reliably, consistently. A key thing to understand about automating your workflows is that it's going to save you reagents in the long run because of the consistency and reliability. There's less uncertainty whether something actually was set up correctly if you've used a robot. So both in sort of a very large overview level and, at least in the Mosquito's case, like practical level, it miniaturizes and saves the amounts of materials you're using for your crystallography workflows.
Perfect, thank you, Hugh. Another question just come in. What is the volume range of the Mosquito?
Good question. Sorry, I didn't explicitly answer that because we were talking about drop sizes. But the volume range of the Mosquito is 25 nanoliters up to 1.2 microliters in one aspiration and dispense.
I can add on to that.
Yeah.
So I've used it as low as 50 nanoliters where protein has been limiting and actually being able to successfully get hits as well. I wouldn't recommend it. Of course, if you're using it at low volumes, I would highly recommend the humidity chamber as well, because it keeps in a very controlled environment, and that is important.
Actually, that's a good point. The humidity chamber is a part of the Mosquito Crystal 3 package. We generally show it without it because it's hard to see what's going on with it on in these picture formats. But yes, this humidity chamber is able to keep the relative humidity between 70 and 80%, ensuring that your small drops evaporate much more slowly and allowing you to set up multiple drops in a well more consistently.
Thank you. Next person asks, what applications can these instruments be used for beyond crystallography?
So that's a good question. The main applications that can be used for these instruments beyond crystallography. So the Apricot S3 is extremely versatile for plate stamping, for serial dilution. It can be excellent for reformatting between plates. So if you want to take from a 96 well plate and stamp out into a 384, it's very easy for that. It also can do the reverse and pool 384 samples into a 96 very easily. Because of those functions, it's very applicable to a huge range of applications in genomics, in basically any kind of biological lab. The Mosquito, at least the low volume one that dispenses and aspirates 25 nanoliters to 1.2 microliters, has seen use in crystallography, but it's also seen use in high throughput screening and drug discovery labs, where it is used for serial dilutions of compounds and creating miniaturized assay plates so that you have concentrated compound in miniaturized amounts of DMSO that can go into your cellular assays. It also has found use for certain kits in genomics, which require extensive miniaturization to keep them affordable for that particular lab. The Dragonfly family of instruments has been used across all sorts of different things. It's one of the most versatile and easy to use instruments we have. Being able to assemble a plate, the Dragonfly is able to create plates not just for crystallography, but for high throughput screening. Because of its liquid agnosticism, it's able to dispense things like PCR master mixes very easily and enzymatic and glycerol mixes. So it's found use in all sorts of sciences, with all types of liquid in the lab. So the Apricot and Dragonfly are excellent walk-up, easy to use instruments. They're very simple. And Mosquito's miniaturization has found a lot of use across a wide range of applications.
Great. Do these instruments remain clean and sterile enough to work with RNA?
Yes, all of them have disposable and single-use, can be single-use tips, making sure that contamination is minimized. From personal experience, I can attest that the Mosquito is excellent at setting up RNA crystal trials. It is clean enough and RNA free enough to use RNA and crystallography reliably. The Apricot tips can come filtered and sterile to make sure that your transfers with those are very reliable. Another thing about the Apricot head is it uses this sort of elastomer gasket seal inside the head, which the tips attach to. that is very easily replaced. This is helpful because unlike certain air pipetters, the mechanism doesn't really get dirty, and you can replace this gasket seal if somehow liquid does get up there, so it's very easy to keep it functional and clean. Also, the open deck of the Apricot makes it extremely easy to wipe down. All three of these instruments have been used in hoods and biosafety cabinets. The Dragonfly, because of the disposable syringes and the fact that the pistons don't make contact with the liquid, keeps it very clean. It's very easy to keep everything sterile on that as well. So all three of these instruments have seen great use keeping in sterile environments and succeeded. One other thing, sorry, to mention about the Mosquito. In the picture shown, it's not shown here, but there is a spool cover option, which is a hood that goes over the spool, ensuring that your tips remain covered and dust-free from use right until they come out of the machine, after which they're used. It doesn't matter. Yes, these instruments remain sterile enough to work with RNA, many other applications that require sterility, such as cell work.
Perfect, thank you. Another question's just come in asking, do you use any reagents such as sodium azide for long storage crystallization screens?
Sorry, could you repeat the question?
Yeah, sorry. They're asking, do you use any reagent such as sodium azide for long storage crystallization screens?
SPT doesn't really make crystal screens, so it depends exactly on the screen in question. Between uses though, you can foil seal or plastic seal on your top of your crystal screens or the plates you've made to keep them clean. We recommend, of course, also spinning down these plates before they are used. But yeah, the exact crystal, it depends exactly on what crystal screens you are using. And although I've mentioned Hampton a few times, there are many other commercial ones that are available. So that would be a question more for the specific manufacturer of that crystal screen.
In practice, I can add that we have not used, I've not seen azide being added anywhere to these screens too.
So I'm just going to quickly recap for everyone. Thanks, Susmith, again for your input. But I just want to recap exactly what these three instruments work together for. The Apricot S3 is for stamping out your plates to get ready for drop setting on the Mosquito. The Mosquito is for reproducibly and reliably miniaturizing your crystal drop setting assays, whether they be sitting drop, handling drop, bicell, or many other types of optimization assay. And the Dragonfly is for creating your custom plates. So although I've talked about crystallography today, there's other applications. Instruments are great for reproducing results from crystal trials and assays that you might fail to do so by hand. They increase throughput and productivity, and they're very easy to program and change on the fly. They generate robust and reliable data. They are fantastic. If you set up control trials with your Mosquito, you can know for sure that your crystallography process is working, so that when you don't get crystal hits, you know it's not just because you pipetted something wrong. And they save a lot of reagents and precious material in the long run, and they're accurate even for high viscosity liquids. Even the Apricot is a lot of good options for dealing with different liquid classes. So even though it's not positive displacement, the Apricot is still pretty robust at dealing with new liquids.
Perfect, thank you both. Unfortunately, that's all we have time for today, but please join me in thanking Hugh and Susmith for their time today. As you leave the webinar, a survey will appear on the screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. So on behalf of Drug Target Review and SPT LabTech, I'd like to thank you all for attending today's webinar. We hope to see you again soon.
Thank you, guys.